I'm leading up to a theory about how evolution produces some of these abilities, and it was developed in collaboration with Jackie Chappell. And the theory is, I'm leaving out some of the details, that human and other animal genomes have a feature which we call which makes us call them meta-configured genomes. What this means is that a genome doesn't just specify everything that's to be done at the start of the life of an organism and then how all the things that are created internally will go on developing. It may do different things at different stages of development, including starting to grow new things at certain stages. That should be very obvious from, for instance, the genetic effects of sexual development in humans. There are lots of physical changes and motivational changes and changes in interest that occur quite late in life, and if they occurred in newborn babies, that would be a complete waste of time. But that's just one example of staggered gene expression. And I think there's a lot more of it going on in different stages, and I'll try to illustrate that with this diagram. The idea is that you have this genetic structure which is up here, and this is capable of producing something in the developing organism, and just producing different things at different times. And what it does at the early stages produces changes internally, and then there might be behaviors that affect the environment, and there might be sensors which get information from the environment after these simple behaviors, perhaps like the ones that I mentioned earlier with little microbes wiggling around. These simpler behaviors can provide information that may be stored. It may be made use of immediately, or it may just be stored because it will be useful later. Then at a later stage, something new might come out of the genome which builds some new structure or some new behavior, which generates something a bit more complicated, perhaps more abstract, and that in turn can influence simpler behaviors and organize them to influence the behavior, to influence the environment, and then there'll be sensory information which will lead to more records. And this can happen in several waves of increasingly sophisticated genetic specifications producing not directly behaviors, but producing information processing mechanisms, producing new contents, like new motives for information processing mechanisms. And when that happens, precisely how the gene is expressed may depend on the records that were previously acquired from much earlier gene expression. And the most obvious example of that is human language development, where initially the genes get a very young child to start babbling, and the child babbles and it may listen to the talking that's going on around it, and it gets influenced in the kinds of muscular movements and these nervous control mechanisms required to produce the sounds that are in some ways related to the ones that it hears. Then later on, the genome can generate new competences and new motives to combine these sounds in more complicated ways, 2 or 3 at a time. And that can happen in the presence of adults who respond, and the child may keep records of what the responses are with these different things, and those records may stay around and be used at a later stage. Now, I don't want to go into all the details of that a theoretical linguist would, which would take up the whole lecture, but the point is that more and more complex information structures can come into records that are acquired by interacting with the environment in more and more complex ways that are produced by later, probably more recently evolved genetic structures, which may have various kinds of abstraction in them. By abstraction, what I mean is that they have details that are not provided in the genome as to what needs to be done, what should be done. Instead, some of the details are picked up from the previous records that were produced as a result of earlier gene expression. So that allows the later expression of a genome in one environment to be very different from the same expression, from the later expression in another environment of the same level of genetic structure. And that is shown very clearly in the case of languages by humans who presumably have roughly the same genome, end up talking completely different languages in different parts of the world, and they will have different stages, and at each stage they will have they may have different sounds, different patterns of sound combinations, different syntactic structures, different semantic contents, and so on, and even different purposes for which they use a language. Now, I want to show an example of what happens when children learn languages, but learn sign languages. And oh dear, something's gone wrong here. Sorry, I went to the wrong place. Okay, this is a YouTube video called The Birth of a New Sign Language in Nicaragua. Some people viewing this may already know about it. For a while, the Nicaraguans thought that deaf children should learn to lip read, because there's no use learning a sign language because the people around them who are not deaf won't be able to understand the sign language. However, learning to lip read is not at all a successful strategy, and they were persuaded by experts that they should teach the children a sign language. So they got a lot of children together, and they had a teacher, and the teacher had been born deaf and had learned sign language, and his job was to teach the students. But very strange things happened, and this is late in the video. I'm going to start it up.
It brought deaf village children into Managua to end their isolation. Here, educators tried to teach them an existing sign language. The effort failed. The children showed little interest in learning a language.
So what happened was that they started signing themselves. They invented their own language. And the teacher had no idea what was going on. There they were, waving their fingers and hands around, smiling, nodding, moving their heads. And eventually they brought in experts from MIT and other places who came and studied these children and worked out that the language they were being taught was much too limited for them, because the poor teacher who had been brought in had never learned a sophisticated sign language. So together they developed their own sign language, and in fact it grew into an independent language. The point about that is that the genome that I was showing you previously, which these children had the ability to make them start doing things which, when they were in the presence of other children who were doing similar things, caused later parts of their gene expression to interact with the results of the early ones, and gradually they were able to cooperate in creating a new sign language. But normally when a young child is developing its spoken language, although it too isn't learning but cooperating, it's in a minority because there are already a lot of other people who speak. So that child's cooperative development looks like learning because it is so constrained by the fact that it's in a minority and it's cooperating with a large dominant majority. So I'll leave that. If you search for deaf children, sign language, Nicaragua, you can find the video and watch the whole thing. It's a fascinating thing to look at. So what I'm suggesting then is what was going on in there is an example of the sort of thing Jackie and I were talking about with their meta-configured genome.